Well, I'm glad to be here today, and I'm going to talk about some brain imaging of brain signals from subjects that we've studied with correlated brain signals. Now, first of all, I wanted to acknowledge all my collaborators and co-investigators: Liana Stannis from Bastyr University, Layla Kozak, Jeannie Achterberg, Karen Cook, James Lake, Clark Johnson, and we wanted to thank Dean Radin for help with our EEG alpha analysis, which I'll show a little bit later on. So the question is there evidence for correlations between distance intentionality and brain function in recipients of distance intentionality who are tested using fMRI and EEG? Now, for a background, history has shown evidence for a connection between human beings. We all like connections between those we love and those that we work with. And so how are we connected? There could be spiritual connections, emotional connections, sensory connections. Now, all of you have a sensory connection with me right now. So you can see me and I can see you. I have a sensory connection. And then there's energy connections. So all of these have influence on how, my theory is on how the brain works. Okay, so a little bit of background. In my regular work, I do brain imaging of different diseases and different research projects. So one of my biggest projects is to study children with learning disabilities. And so I do a lot of brain imaging of language in children with learning disabilities. But so functional magnetic resonance imaging is a technique for measuring brain activation using blood oxygen level dependence, which is called BOLD. But now fMRI is completely dependent on the water in the brain. So we've heard some talk before about the importance of water, and that's exactly what we're measuring in the brain, is the water signal, which may be influenced by the oxygen, but the real signal is coming from the water. So how is it statistically tested? Is there a possibility of false positives? And I'll go through some of the statistical tests that we use to test for the significance of brain activation that we get. So there's been a blossoming of MRI in its ability to brain scan, and I borrowed some slides from Mark Cohen from UCLA. And so and I'm going to give you a little background on brain imaging, but there's a blossoming of advances in brain imaging techniques that's happened in just like the past 5 to 10 years that's allowed us to measure new things in the brain that we haven't been able to do before. Okay, so brain activation is what happens when you think. So if all of you put up your right hand and all of you go like this with your right hand, and as you're doing this, you are activating your motor cortex on your left side. So it crosses over. So your right hand activates part on your left brain. It crosses over. And so you're activating parts of your motor cortex right up here of your left side of your brain. And this causes a change in CBF, stands for cerebral blood flow. It changes cerebral blood volume, oxygen, and venous oxygen and glucose utilization. So all of these things happen as you change your brain activation. Now it also changes the electrical activity in your brain, which you can measure with electroencephalography techniques. And so the technique with functional MRI is dependent on the fact that we have an influence of the blood oxygen on the tissue signal that we get, even though it's some distant. It could be some distant from the blood vessel. So here's the blood vessel in the brain, and we can pick up signal over here because of the influence of the oxygen on the water in the tissue over here. Okay, so one of the first experiments done with functional MRI was with photic stimulation. And so you can measure the brain signal, say, every 3 seconds. And you keep going beep, 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 and you go on, and then you go off, and you go on, so off and on. And as you change the stimulus, you get a change in the brain activation as seen by the MR signal intensity. And so we can see this change, and then we can do statistics to test, is this significantly different than this for all the different parts of the brain? So in photic stimulation, we, in this case, they're stimulating for 60 seconds at a time, and then they switch tasks for another 60 seconds, and then they switch. Now, typically, we go through about 5 cycles of on-off period to get enough statistics to make a statement about brain activation. So this is an example of activation with a moving visual stimulus. And so here it is in the back of the brain, in the MT region of the brain, V5. These are areas of visual processing in the brain. Okay, and then to process the signal, we do some convolution of the impulse function in order to process this, because there is a delay. So after you get a stimulus, there is a delay of about 5 seconds between the onset of the stimulus and the change in the bold signal. Okay, so the temporal resolution is limiting in fMRI. That's one of the limitations. And that's why we like both fMRI and EEG, because the temporal resolution is very slow in fMRI. But the spatial resolution is very good. So we like to do both fMRI and EEG because we get the best of both worlds, the spatial resolution with fMRI and the temporal resolution with EEG. Okay, now we use software developed by a group in Oxford called FSL. So we have software developed to do the statistics of the brain signal. And we can measure single subject statistics, and we can do group statistics. Oh, let me go. Okay, and it's called the GLM estimation. So we have this model that we apply to the brain signal, which we use to figure out how close the model fits with the actual signal we get from the brain. And we use that model to test every voxel in the brain to see if there's a significant activation. And we do some autocorrelation, and we're able to get a brain activation map in the brain. I'll show you some in a minute. But now we've published a paper in the Journal of Alternative and Complementary Medicine. Jeannie Achterberg is the first author on this paper. She's from Hawaii, and so is Karen. They're both from Hawaii. I'm from Seattle, and my job was to analyze the signal. So they sent me the images that came from the scanner, and my job was to analyze the brain images. So the subjects consisted of 22 participants, 11 pairs of healer and recipients. Now, in the big island of Hawaii, they have these Hawaiian healers that are gifted. These are the gifted Hawaiian healers, and they know how to send their energy, and they know how to influence their energy for good to heal. So Jeannie is the one who selected these healers to do this, experiment with the fMRI. So in the experimental conditions, the healer was in a control room, physically and optically isolated from the receiver and the scanner. So the scanner is deep in this tunnel. If you've ever, how many of you had an MRI? If you had an MRI, it's kind of daunting because you have to go in this deep tunnel. It's really noisy, and you say, how could you do a brain activation like this with all this noise? How could this ever work? Well, it does work because you can you have the on and off stimulus while the noise is going on. So the noise is constant, and you have the on and off stimulus during that time. The healer was verbally instructed with cues to start and stop the distance intentionality in a random pattern. So they were told to start and stop in a random pattern in 12 different time periods. Now the scanner was a 1.5 Tesla. This is a very strong magnetic field, MR imaging system. I might mention that there's some people that told us that they lose their psychic abilities when they go in an MRI scanner because the magnetic field is too strong and they lose their psychic abilities. So I've heard that before. But anyway, with 1.5 MR imaging system, we can get very nice images. And for those of you involved in brain imaging, these will make sense to you if you're involved in brain imaging. These parameters are important to get good contrast in the brain related to the blood oxygen level dependence. So then we did fMRI analysis of the on-off patterns dictated to the healer. We used FSL from Oxford, the UK, to analyze the signal. We have single subject analysis and group analysis. So we do a group analysis after we do the single subject analysis. And then the results show that in the group analysis we had two very significant clusters. Now, let me tell you again what this means. This is a group analysis of the receivers who are inside the scanner who are receiving signals from the healers. So all of the signal comes from the receivers, not the senders. And so the receivers are able to receive the signal from the healers, and we're able to measure the signal in their brain, and we get these two significant clusters with a probability of false positive 8.5 times 10 to the minus ninth. So I think that's significant. That's 10 to the minus ninth is a significant significance. And we had another cluster of a little less significance. And so we can test the probability of using these techniques. Now, this is kind of blurry. I have better images than these. But this is one of the clusters in the back of the brain again, this is the group analysis cluster. And again, this is a part of the brain involved in visual processing and in imagery. So this is a part of the brain involved in image processing, even though they got no stimulus except from the healer. The healer was the only person giving the stimulus. And they got a signal here in the back of the brain. And then we also got a signal in the front of the brain, in the frontal regions. And so the anterior cingulate is involved in attention to stimulus. So that's one of the parts of the brain. And also the frontal regions are involved in a lot of higher order cognitive processing. So we get a lot of higher order level of cognitive processing, including language. So we get a lot of frontal areas involved in language. And so when we map those clusters, these are all the different regions that we get of activation from those group analysis of the clusters. Okay, so here's another paper that we published, again in the Journal of Alternative and Complementary Medicine. And this was one of the first papers we published on just a couple of case reports. So the last details that I showed you involved 11 pairs of subjects, whereas this was only a couple of pairs. And again, we were showed signal in the back of the brain, and here's the time domain correlation of that signal. So we can plot the time course of the brain signal as a function of time as they're being as the person outside the scanner is sending the stimulus. In this case, the sender was getting checkerboard, flickerboard pattern So in this case, it was not a healer. It was just a sender that was watching a checkerboard flicker pattern that was randomly going on and off. And here again, we get the activation in the occipital visual parts of the brain. Okay, now some of these same people also, we did EEG analysis. Now the difference with EEG is that both the sender and the receiver were hooked up to the cap. They all got this cap with 16 channels of EEG recording. And in this case, we could record both the alpha wave pattern of the sender and the receiver. And we published this. Liana Stannis was the first author on this paper. And in this case, we were measuring EEG evidence of correlated signals. And this is the setup that we had at Bastyr University. So we had one subject positioned in one room, and he's watching a visual stimulus. We have the control center, and then we have another subject physically isolated from this other subject. And at one point, only one of the subjects is getting a stimulus, and the other subject is acting as a receiver. So I really like alpha waves because I've actually used alpha wave therapy in some of our patients with multiple sclerosis. So I like alpha waves, and I will show you the results. So Dean Radin suggested a technique for us to use to correlate the difference between the EEG signal between the senders and the receivers. So we used that technique, and we got a regression with a significance of 0.011. So this is a regression analysis between the sender signal and the receiver signal of their alpha. And so when you do a correlation, it's kind of scattered, but there is a significant correlation between the senders and the receivers. So the pink line is kind of the regression line, and all these individual dots is one dot from the sender-receiver pair. So this was a significant effect, correlating signals between the senders and the receivers who were isolated from one another. But now the group, again, the group at Bastyr University helped us to choose these people who claimed to have a connection with their partner. So these people claimed to have a connection. And so some of them are more gifted than others, and they could repeat the effect over and over again, whereas some of them had more of a weak correlation. So just as with everything, you have the gifted and then the semi-gifted people. So these findings support previous research on distance healing, suggesting that human intentions may directly affect others in ways that are not entirely understood. But with functional imaging and with EEG, we can study these effects, and we can measure a signal in their brain. So I'll stop with that.
We have time for quite a few questions.
You mentioned at the beginning of your talk that when you had us raise our right hand, that would have left brain stimulation.
Right.
I had been to a kind of a healing demonstration some years ago with Donna Eden, and she chose people in the audience to demonstrate a principle, and she said they had homolateral, they were homolateral, which I think means your right brain is working with your right hand. And she performed some work on them energetically in very short order. that enabled them to cross over. And so I'm wondering if you have ever heard of something like that or seen anything like that, and if it would show up in the MRI imaging.
Yes, as a matter of fact, one of the experiments we usually do to test the scanner is a finger tapping experiment. And when we do finger tapping on-off, we get activation, and a lot of times we'll get bilateral activation, but it will be stronger on the contralateral hemisphere. But I can understand that there could be some people that have homolateral activation. But the usual case, the usual brain, is for contralateral activation. If you do your right hand, you usually get left-sided activation. That's the usual case. Right.
Todd, do you think that the setup of your EEG experiment, where you had all your monitoring equipment directly in line in between the sender and receiver, may have not given you such good results as if they'd have been separate in a different way?
Yeah, are you asking because they're in line, or?
Yeah, right next. The sender and the receiver are on the same straight line as your electronic monitoring equipment.
Right.
Is that true?
That was pretty much true. They were 3 rooms in a row, so they were 3 rooms in a row, but there was quite a big distance between so that in that diagram they look close together, but there was quite a big distance. Those were pretty big rooms that there were in between them.
I wonder if you might have any speculation or explanation, perhaps, why I believe the Hawaiian situation was healers, why there would be a strong response or a noticeable response deep in the optical region of the brain.
So that's a good question. Why in the optical region? One of my explanations is that there's some imagery involved. So maybe what's happening is that the healers are invoking an imagery process in the receiver. So the truth is, in normal visual experiments, if a human will picture an event and just make an image in his mind, the brain activates as if he's really seeing the picture. So it's a very close coupling in the brain between reality and just thinking about a picture. So if you think of a face, you'll activate the fusiform as if you were really looking at a face, even though you're just thinking about a face. So I think it's the imagery that's being evoked by the healer. That's one of my theories.
I'm curious how the sender identifies the receiver. Not just in your talk, but in previous ones as well. But in some cases, clearly you have them connected by circuitry. In other cases, they're in separate rooms. So I can imagine the sender would know the person's name, perhaps, would know the location, might be thinking geospatially as to, oh, a person located there, or might be thinking, oh, Fred. What is the sender doing mentally to identify the receiver?
Well, that's a good question. Before they started the experiment, we had the healer spend some time with the sender. So they spent some time together, getting to know each other, and so that the sender really did have a good impression about who the receiver was. So they did spend some time together before they separated. But we've done it both ways. If the sender doesn't know the receiver, it doesn't work as well. It almost doesn't work nearly as well as when the sender knows pretty well the receiver. Does that answer your question?
Sorry, I'm having a bit of deja vu here and wishing I could get at my reference library, because I seem to remember, but I'm blocking on details, that quite recently there's been another fMRI study that garnered some publicity for claiming to have refuted the whole ESP concept once and for all and showing no correlation between senders and receivers in an ESP task.
Really? Do you know who the author was?
That's why I wish I could get at my library to check.
Oh, really? Well, I can picture an environment when nothing would work. You can sabotage the environment and make it so nothing works. I mean, several people here have mentioned that. The environment is very important in how you set up the experiment. And so I think that the environmental impact can influence how well the healer can send to the receiver. So I can understand how there could be a false result.
I'm curious, in what way were the alpha waves linked? Do they have the same frequency? Were they synchronized, or how did that work?
The alpha waves were linked in the way that they differed between the flicker on state and the flicker off state. So we were comparing the alpha amplitude between on and off. And that distance and that difference between those two states is what we use to correlate. Does that,
amplitude, okay.
the amplitude of the alpha between the flicker on and the off state.
I had a question about the receivers. I mean, who were they? What were they told? Did they have a background of meditation, or were they involved with the healers, or are they just some people off the street that they grabbed? The receivers.
The receivers.
Yeah.
The receivers were chosen, as far as I know, by the healer, but I'd have to ask Jeannie that question. I'm not entirely sure how they were chosen, but I do know that they did spend time with the healer before they went in the scanner, so that the healer did have a really good relationship with them before they went into the scanner.
And imaging then is sort of part of this healer's idea, so they could be transmitted to the receiver if they talk to each other?
Wait, what's your question?
Well, if the healers are into imaging, then if they talked it over with the receivers, then that may help explain why it's you're seeing it in the imaging part of the brain.
Oh, I see what you mean. I see what you mean.
Okay. I'm going to let
Yeah.
him speak, and then you're next. Because it's always
Todd, that was a great presentation. Thank you.
Thank you.
I happen to have talked to Jeannie Ochterberg about how those subjects were chosen, and she says that the healers were asked to recruit a subject with whom they had worked in the past and had some sort of empathic emotional connection. And so the empathic component to the whole deal seems to be really crucial about what you found. And it seems to me that these large-scale double-blind healing studies where you have typically strangers praying for strangers completely subvert one of the necessary factors, at least in your study, for a successful study.
Right.
So that's probably a great shortcoming of the double-blind randomized study as it's done in many places.
Right. That's so important for the healer and the receiver to have a good relationship and empathic, emotional type of connection.
And in the follow-up study that you all did, I understand that that empathic bond simply wasn't there because these, in the rerun of the study, the healers did not know the subjects.
There was one where Jeannie did one where they didn't know each other.
Yeah,
Is that right?
Yeah.
And
so,
that showed no significance.
It was a flop.
It was a flop. Yeah. So that makes sense to me why this other person got a negative result if they didn't set it up, a good connection between the healer and the receiver. That's so crucial. And I know you do that with your work too, right? You establish a good connection, right, in your work. Yeah.
Could you talk about the control aspects of this on the first experiment? In other words, there was the sender would set up a connection with the recipient, and then the sender would not send at certain times,
Right.
and there would be no response.
Right. So it was on, off in a random manner. So the manner was randomly on and off. So in fMRI statistics, you do this statistics that says, okay, I'm going to test a part of the brain, and I'm going to test whether there was a significant correlation with the stimulus. So the control, in essence, is the off period and the on, and the randomization of this on-off condition.
You're working here with distance healing, all very positive. Now, certainly in folklore, there's a long history of negative influence at a distance. Have they done anything in looking at the ability to adversely affect a subject?
Adversely affect.
Well, and if you read folklore throughout the world, we were talking about curanderos this morning, they will argue 50% of their work is in the negative domain.
Yeah, I've heard about that. And I know scientists that are worried that some of the intentionality will get in the wrong hands of somebody wanting to do harm. I'm more interested in healing, but I do know of some scientists who have told me of experiments done with the intent to do damage and instead of the healing part. I've heard of those.
One of the variables you're speaking of is the previous emotional connection. I'm wondering whether or not there's any possibility of looking at the data in terms of whether the healee had a need for healing. So do those people who are in need of healing, even if they've had a previous emotional bond with the healer, as they become more healed, or let's say the healing process is complete, does that weaken the resonant phenomenon?
I don't think we've studied that, but that's a good question. We haven't studied that aspect of it, but there's a lot of aspects of the healer and the emotional aspects of the healer and the receiver and their health that could impact it. I can see how that could impact it, but we haven't studied that yet.
This has to be the last question. I'm sorry, but no. Oh, that was the last question. Yeah, it is the last question,
Okay.
and